Okay, this is question 2 from Key Assessment 1. Lovely, lovely. Mitosis and meiosis are both forms of nuclear division. They are. Both types of nuclear division are involved in the production of gametes. Image 1 shows a section through part of an anther of, I might just, so what are we looking at? We're looking at an anther, so I would be thinking now this is making the cells at the start, so making more of them, and then with mitosis I would also be thinking, oh, hang on a minute, then we've got that sort of generative and tube, and then we've got the generative also splitting by mitosis. And meiosis, of course, is reducing the chromosome number. And we start with 1 cell, we go to 2 cells, we go to 4 cells. So this is 2n, that's n, 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 n. So we're showing stages in the production of pollen grains. What have we got? We've got the tapetum layer. That's the epidermis. We're looking at 1 pollen sac. And we've got 3 sorts of cells. So this one's got 2 nuclei. This one's got 4, that's our tetrad. And then these are the tapetum. And I'm thinking they must be diploid. Okay, so it was concluded that structure A had just completed meiosis 1. That's the one with 2 nuclei. And structure B had just completed meiosis 2. Explain how you would draw these conclusions from the micrograph. So meiosis 1 gives rise to 2 cells, shown by A, and meiosis 2 gives rise to 4 cells, and B has 4 nuclei. Can't think you could say anything else from that. Cells labelled C are in interphase. Okay. Explain why nuclei are only visible in some cells. Are they? Oh yeah, that one's not got one. Oh, neither is that one or that one. Where nuclei are visible, why they appear to be of different sizes. Okay, so the section may not have been taken through the nucleus of some cells. So they look like they haven't got one. Remember, cells sort of 3D like that. And if you're cutting through it, you could cut through it in the middle and there might be a handy nucleus there, but what if it was down at the bottom? You wouldn't see that at all, you'd just be cutting through. Why would they be different sizes? So if they are cut in different planes, the nuclei would appear differently. If you think the nucleus is there and you cut it right through the middle, it's going to be big. If you cut it up there, it's going to look smaller. If you manage to cut it, you know, at some weird angle, it'll look elliptical. Deduce whether this cell, alright, hold on a minute, so you can see it as well, that'd be handy, wouldn't it? drawing was made of an animal cell during anaphase, so hence the spindle. The diploid number in this cell is 8, so we've got 2n equals 8. So these are chromatids, and so we've got a pair of, so that would be a chromosome, 1, 2, 3, 4. So we've only got four chromosomes there. So this is a haploid cell. So just going back to meiosis, we start off, we've got our homologous pairs. Then you've got two cells, and they've got one on each spindle, and then they separate like that. And that would be meiosis. And if it was mitosis, but it can't because we already established it's haploid, we would have our homologous pairs like that, and then they would split, and then you get two identical cells, wouldn't you, with two chromosomes in. So, deduce whether this cell is undergoing anaphase of mitosis, anaphase 1 of meiosis, or anaphase 2. We're going to use this diagram to explain why it's not mitosis, why it's not anaphase 1, and why it is anaphase 2. So, the cell is at anaphase 2 of meiosis because the cell is haploid, it only has 4 chromosomes, if it was anaphase 1, oh, can't write and talk. If it was anaphase 1, the homologous pairs would be separating. And if it was mitosis, then there would be 8 chromosomes. I think that will do, really. Okay. Well, that was mercifully short.